We're starting the entire curriculum with the cell membrane rather than something more obvious like the brain itself. Now, every single thing a neuron does depends on this structure, this cell membrane structure. This could be generating an electrical signal, propagating an action potential down an axon, releasing neurotransmitters at a synapse, or among countless other activities. Now, if you memorize facts about these ion channels and action potentials later without a mechanistic understanding of why, you won't truly understand the later content. Now we have 8 lessons to cover in this lecture. My goal is to cover approximately 30 minutes of material in each video. So some videos may include more lessons than others. Personally, when I study, I find it helpful to work through smaller chunks at a time. So I've divided this lecture into 8 manageable sections to make it easier to study. Now, before we can talk about anything crossing the membrane, we need to nail down what each membrane actually is and what's stuck inside it. Now, this first section will cover the bilayer structure, fluidity, and membrane proteins. Now, a lipid is any biological molecule that does not dissolve well in water. This definition is inherently based on behavior as opposed to structure like some other definitions are. Now, to understand why, we need to talk about water itself. Water is a polar molecule, so its electrons aren't evenly shared. So they are not evenly shared between the oxygen and the hydrogen atoms. So it has a partial negative charge on the oxygen side and a partial positive charge on the hydrogen side. That charge separation between the hydrogen and the oxygen is what lets water molecules form hydrogen bonds with each other and with other polar or charged species. Now, lipids, on the other hand, are composed of these long chains of hydrogens and carbons that are called hydrocarbon chains or fatty acid chains. Carbon and hydrogen shared electrons almost perfectly evenly, so there is no meaningful charge separation between the two, which also means there's no hydrogen bonding capacity and no attraction to water. And that's pretty much the entire chemical basis of this concept of hydrophobicity and why, for example, oil and water don't mix. And lipids are hydrophobic for exactly this reason. Now let's talk about the specific lipid that makes up our cell membranes, the phospholipid. The name tells you the structure exactly. So phospho is in reference to that phosphate group and lipid for the hydrophobic tail region. Now the head of the molecule, this round circle here, is the phosphate group. Now that's an atom of phosphorus that's bonded to several oxygen atoms. Now this phosphate group carries a negative charge. Because it's charged in polar, it's also hydrophilic. Hydrophilic comes from the Greek meaning water, hydro meaning water, and philic meaning loving. Now the phosphate head is water loving, so hydrophilic. The tail is 1 or 2 fatty acid chains. In this picture, it's 2 fatty acid chains, which have no charge. So the tail is hydrophobic and disrupts water's own hydrogen bonding network if you force it to come into contact with water. So these fatty acid chains actively avoid water, and that's what we would call hydrophobic, coming from the Greek hydro, meaning water, or phobic, meaning fearing. So these fatty acid chains are water fearing. Now, a molecule with one region that loves water and one region that hates it is called amphipathic. This comes from the Greek amphi, meaning both, or pathos, meaning feeling. Now, if I were to drop a large population of phospholipid molecules into, say, a glass of water, they would immediately rearrange themselves. This is a process called self-assembly, which requires no enzyme, no energy, no cellular signal, nothing like that. Now, this self-assembly process happens because of thermodynamics. The system wants to find its lowest energy configuration. So what is this low energy state or this lowest energy configuration? It's when we have heads, these polar heads facing outward toward the water on both sides, with the tails clustering together in the middle, being shielded from the water by the polar heads. This is called a lipid bilayer. So the ends meet tail to tail in the hydrophobic core, being sandwiched between the two hydrophilic surfaces on the outside. This is the fundamental structure for every membrane in the body, from the plasma membrane to organelle membranes to axon membranes to dendritic membranes, all of it. So we have this barrier property that emerges automatically. There is no wall building machinery required. This barrier is why the inside of a cell can be completely chemically different from the outside of the cell. And it is also the physical basis for how a neuron generates an electrical signal, which we will talk about later. Now, real membranes aren't static walls. They are very dynamic, and the framework we use to describe that is the fluid mosaic model, which was proposed in 1972, and it's still the foundational description that we use today. Now, fluid means the individual phospholipids are not locked into place, so they drift and slide laterally within their own layer constantly at normal body temperature. Now, we will discuss some processes later like vesicle fusion and receptor clustering that absolutely depend on the membrane behaving like a two-dimensional liquid rather than them just being frozen solid. So that fluid state is very important to neuronal physiology. Now, mosaic means the membrane is a patchwork. It's made up of a bunch of different proteins and other molecules that are embedded within the lipid C, kind of like you would see tiles in a mosaic artwork. And then here we can see that here. We've got all sorts of proteins and channels and carbohydrates. So we got protein channels, globular proteins, glycoproteins, carbohydrates. We can see it's pointing to our hydrophilic heads, the phospholipid bilayer itself. There are alpha helix proteins, cytoskeletal filaments. All of that makes up that mosaic patchwork. Now, let's move into lesson 2, where we ask fluid compared to what, and what actually dials that fluidity up and down. And we have 4 main variables with this: tail length, tail saturation, cholesterol, and temperature. All right, starting with tail length. Longer fatty acid tails have more surface area in contact with a neighboring tail. Now, this means that there are more van der Waals interactions occurring. Now, just a little bit of review, what are van der Waals forces or van der Waals interactions? These are very weak, short-range attractions that sometimes happen from transient fleeting shifts in electron distribution, which creates a brief small change of charge imbalances. Now, individually, these van der Waals interactions are very tiny, almost negligible, but when there are a bunch of them happening at once, they can accumulate along the length of a chain. Now, the longer chain means more atoms lined up next to the neighbor, more cumulative attraction, tighter packing, and therefore decreased fluidity. So shorter tails means less contact surface, weaker cumulative attraction, and increased fluidity. So in summary, longer chains decrease fluidity and vice versa. So shorter chains increase fluidity. Our second variable is saturation. A saturated fatty acid has every carbon bonded to the maximum number of hydrogens possible, so all single bonded along the chain. Now, these single bonds allow free rotation, so the chain adopts a long, straight, rigid conformation. So you can kind of think about an uncooked strand of spaghetti. An unsaturated fatty acid has at least one double bond somewhere in the chain. Now, a double bond does not allow free rotation, and in the vast majority of biological fatty acids, it occurs in the cis conformation, meaning that the 2 chain segments on either side sit on the same side of the double bond, which produces a fixed kink, kind of like a bent elbow. Now, if there are multiple double bonds, it's what we would call polyunsaturated. You get multiple kinks. Now, the more kinked a chain is, the harder it is for the fatty acids to be tightly packed to their neighbors. So looser packing means weaker van der Waals attraction and increased fluidity. So you can observe this kind of like in your kitchen, for example. So butter, that's mostly saturated straight chains, so it's solid at room temperature. Olive oil, on the other hand, is mostly unsaturated and they're kinked, so it stays liquid because those chains just can't pack together tightly enough to solidify. So, cholesterol. Now, I personally think that cholesterol is probably the most interesting of our four variables because its effect isn't one directional, it's a buffer, it kind of goes both ways. So structurally, cholesterol is a steroid. It's a rigid backbone of four fused carbon rings with a single hydroxyl group at one end and a short, flexible hydrocarbon chain at the other. Now that hydroxyl group is polar, so the oxygens are pulling electron towards itself, like we talked about earlier with water, making the small region hydrophilic, just like a phosphate head. Now the rigid ring system and the short tail are hydrophobic, so the cholesterol, like the phospholipid itself, is amphipathic, meaning we've got part of it that's hydrophilic and part of it that's hydrophobic. Now, the cholesterol inserts itself into the bilayer accordingly, with the hydroxyl group oriented toward the same surface as the phosphate heads. So the cholesterol also points hydrophilic to hydrophilic, and then the hydrophobic region, so those rings sit parallel to the hydrophobic fatty acid tails in the hydrophobic core. Now, at high temperatures, when tails have a lot of thermal energy and are moving vigorously, the cholesterol's rigid rings act like a stiffening rod, restricting the motion of the surrounding tails. So cholesterol decreases fluidity in this condition. Now, at a low temperature, when tails want to settle into tight ordered packing, cholesterol is really bulky and it's got this irregular, odd shape, and it kind of gets wedged between the tails, and it physically prevents that tight packing. So cholesterol increases the fluidity in this condition. Now, the net effect across a range of temperatures is that cholesterol narrows the range of fluidity the membrane can have. So it functions as a buffer on either side. So when temperature gets too high, the cholesterol restricts tail movement, so it decreases fluidity. And when the temperature gets really low, cholesterol prevents that tight packing, which increases fluidity. Now let's talk about temperature. As temperature drops, tails lose kinetic energy and they move more slowly, and therefore those van der Waals attractions pull them into progressively tighter packing. Now, if you drop the temperature far enough, and a pure phospholipid bilayer, meaning no cholesterol, can actually undergo a phase transition, shifting from a fluid or a disordered liquid crystalline state into a rigid, tightly packed gel state where lateral movement essentially stops. Now, the specific temperature at which this happens is called the transition temperature, but it isn't universal. It depends on the fatty acid composition. So not all of these lipids are the same, so the temperature at which it transitions is different depending on the length of the fatty acids. Now, longer, more saturated tails raise the transition temperature higher, while shorter, more unsaturated tails lower it. Now, cholesterol's role here is to soften this transition. So rather than the membrane abruptly snapping from fluid to gel at once, the cholesterol widens the transition, which allows it to have a gradual change, and it's a bit more predictable with the cholesterol. Now, we haven't touched on ion channels yet, but when we get there, you'll see that proteins need a specific fluidity range to fold correctly and to migrate where they're needed. Also to undergo their own conformational changes during function. A membrane that's too rigid or too disordered directly impairs protein function, which is exactly why cells actively regulate saturation and cholesterol content. Now into the next lesson. We need a working vocabulary before we can actually talk about membrane proteins specifically. A protein is built from amino acids, each with the same core structure, a central carbon bonded to an amino group, a carboxyl group, a hydrogen, and a distinct side chain called an R group that gives each of the 20 amino acids its own chemical personality. Now, amino acids link together through peptide bonds, forming one amino acid's carboxyl group and the next one's amino group, releasing water into the process. Now, that chain is a polypeptide. The specific sequence is called the primary structure. One especially important folding pattern is the alpha helix. Now, the alpha helix, we have two types of secondary structures. That's the alpha helix and the beta helices, or the beta pleated sheets. Now, alpha helices are a stretch of chain that coils into a tight, regular spiral, and these are held together by hydrogen bonds that form at regular intervals along that backbone. Now, the reason a protein folds this way at all traces back to the exact same logic we've used all lesson. The hydrophilic side chains face outward toward the water, and the hydrophobic side chains cluster inward away from the water. Now we can classify membrane proteins by how they associate with the bilayer. An integral membrane protein sits embedded directly within the lipid bilayer, with at least part of it inside the hydrophobic core. To remove one of these, you need to disrupt the bilayer itself because the protein is physically anchored in the hydrophobic environment. And you would typically do this with a detergent that dissolves lipids. So why can it sit there and so stably when we just said that hydrophilic things are excluded from the core? Now, this is because the specific stretch of the protein sitting in the membrane is predominantly from hydrophobic amino acids. So they are more chemically comfortable surrounded by nonpolar fatty acid tails for exactly the same reason that the tails themselves are comfortable there. Now, peripheral membrane protein never enters the hydrophobic core at all. It associates with the surface or with the exposed hydrophilic portions of integral proteins through weaker electrostatic attractions or hydrogen bonds. Because these interactions are weaker and don't require disrupting the bilayer, peripheral proteins come off with much gentler treatment, like a change in salt concentration or pH. One more category worth naming, a lipid-anchored protein is technically peripheral. Now, the protein itself never enters the core, but it is covalently attached to a separate lipid molecule that does insert into the bilayer, which tethers an otherwise fully hydrophilic protein to the membrane surface. Now, a stretch of a protein's amino acid chain that spans the entire hydrophobic core from one surface to the other is called a transmembrane domain. It's essentially always folded into an alpha helix, and here's why that matters mechanistically. The regular repeating hydrogen bonds holding the helix together satisfy the backbone's own polar bonding requirements internally. So the helix doesn't need to interact with water along its backbone at all while sitting in the nonpolar core. Only the side chains facing outward into the lipids need to be hydrophobic. A single pass protein that crosses the membrane once, one end, the N terminus outside the cell, and the other, the C terminus inside, or vice versa. A multi-pass protein weaves back and forth across the bilayer multiple times, alternating hydrophobic stretches embedded into the core with hydrophilic loops sticking out into the water on either side. Now, to keep this multi-pass architecture in mind, it's going to become, I would keep this multi-pass architecture in mind because it's going to become essential when we get to the ion channels, which typically arrange multiple transmembrane segments around a central pore. Okay, I think we're going to wrap up this first lecture video here in the next couple slides just because we are getting kind of close to the 20-minute mark, and I want to keep these pretty short. So, glycosylation, which is the covalent attachment of a carbohydrate chain, usually an oligosaccharide, to a protein or lipid. Now, if you attach this oligosaccharide to a protein, you get a glycoprotein. If you attach it to a lipid, usually a phospholipid, in the outer leaflet, you get a glycolipid. Now, this happens inside the endoplasmic reticulum and the Golgi apparatus, and we will cover those organelles properly in a later lesson, but for now, just know the protein passes through these compartments during construction, where the enzymes attach specific sugar chains before it ever reaches the cell surface. There are two chemical patterns. The first is N-linked glycosylation, or those N-glycans, that attaches the sugar to the nitrogen in asparagine side chain, and then O-linked, that those O-glycans right there, the O-links that attach it to the oxygen in either serine or threonine's side chain. Something important here, glycosylation occurs almost exclusively on the extracellular facing surface, so the outside of the cell, almost never on the cytoplasmic side. Not saying that it's impossible or that it just doesn't happen. It's just that usually these molecules are oriented outward during processing. So it does occasionally happen. Sometimes it happens intentionally, but for the most part, we'll say that this is an extracellular process. All right, this will be our last slide for this lecture. Now, the cumulative sugar coating across the entire cell surface is called the glycolac. So all of those N and O glycans from the last slide, we call that glycolax. Now, this literally means sugar coat. Now, it functions as a molecular identification barcode for cell recognition, and it provides physical and chemical protection. It also participates directly in cell signaling, since many of these receptors depend on their attached sugar chains to be able to bind properly. Now, in the nervous system specifically, this plays a role in how developing neurons recognize appropriate targets while forming connections, and the specific glycosylation patterns influence how certain ion channels and receptors behave once inserted into the membrane. So the whole point of this glycolax, each cell, or each type of cell rather, has its own glycolax, its unique glycolax, which would be similar to how all human people have unique fingerprinting.